Thank you, Dennis, for the introduction and for inviting me to your program. So my name is Claudio Reggiani. I am a PhD student at the Université Libre de Bruxelles, which is based in Brussels, Belgium. And my desk is actually at the Inter-University Institute of Bioinformatics in Brussels, which is a joint effort between two Brussels universities, the ULB and the VUB, to perform bioinformatics research. So I will start giving a broad concept of this research. This research has been published in Genome Medicine in July, and it lies at the intersection of three broad domains, that is biology, computer science, and clinics. Now, the research started with a clinical question, and we wanted to use computer science tools or pipelines, bioinformatics, to tackle these questions. Along the way, we find out some biological knowledge, new biological knowledge that helped us to answer the original clinical question. So this is the framework on which the research has been performed. I will move on to provide a little bit of background in biology and a little bit of background in clinics. So let's start with the DNA and the gene. Begin the place where the transcription machinery binds to the DNA to start its job. In this case, to produce an RNA out of 5 exons. Then this RNA is translated into a protein. Now, gene might have multiple transcription starting sites. That means multiple RNAs may be produced, and therefore proteins with different functions are produced in the cell. Now, what is the point of this slide? The point is, if for some mutation you don't have the first exon because it is because of some event, then all the downstream byproducts are also missing in the cell. And this is usually not a good news for your cell, because proteins have a function in your cell. And if this happens to be a gene expressed in the brain, then you have some malfunctioning on your brain. Could be, for instance, a problem in the growth of the cell or of the development of the cell into a fully functional neuron. Now let's go back to the DNA, to the gene structure, specifically to the beginning of the gene, so the transcription starting site, and its upstream region that is called promoter. This is the place where transcription factors can bind, thanks to some DNA sequences that are really attractive for their perspective. This helps the recruitment of the RNA polymerase, which actually starts the job of the translation, from the transcription starting site till the end. Now, I would like to make the point here that transcription factors and RNA polymerase need space to bind to the DNA. I tell you that because the DNA may be in a closed status. That means that means the DNA is wrapped around proteins that are called histones. And because of that, there is no space for other proteins to bind nearby. Now, there are some signals that help the DNA to unbind from the histones. The one that I would like to focus is the epigenetic factor H3K4 trimethylation. That is the trimethylation of the fourth amino acid in the histone number 3 that happens to be a lysine. Now, this is very important for our research because it is a signal to open the chromatin first, but also in literature is associated with promoter activity. And because of that, because of this epigenetic factor, there is enough space for the proteins, that the transcription factor and RNA polymerase, to bind and do their job. Now we'll provide a little bit of background in clinics. And what I need to introduce is what is a copy number variation. And for this, I will show what is a chromosome. In this case, this is stereotyped as with this figure, and the chromosome is segmented in 6 parts, from A to F. Now, if for any event you miss one of the segments, then you have a copy number deletion. In this example, the D region is missing. So you have a copy number deletion of the D region. On the other hand, you could have a copy number duplication. In this case, the D region is present twice in your genome, could be even 3 or more times. In the context of our research, we are mainly interested in deletions of regions. Okay, final slide on the background section is a little description of the DLG2 gene. This is a gene known to be expressed in brain, and I would like to report in this slide the concept described in a Nature Neuroscience of 2013 that studied exactly this gene and concluded that this gene is associated with complex learning, cognitive flexibility, and attention. Now, the background section is completed. I'm happy to introduce what I've done for this research. So, our work started with the DLG2 gene, and here is represented with this box with the multiple exons. DLG2 gene has 34 exons, and I provide below the isoform structure plus the known proteins that are present in UniProt, SwissProt. Those proteins are named H1, H2 till H5. Now, our interest in DLG2 gene started at this point because the medical doctor I'm working with was collecting patients from the local database of patients from ULB, from the public database of the Decipher, and from literature. And what happened is that the patient that the medical doctor collected happened to be all in the middle of the gene, specifically between exon number 7 and exon number 9. Now, the copy number variation that you see in this slide are all deletions. So they are all copy number deletions. If we do some counting about these copy numbers, we have a total of 29 patients in this region. 24 of them have neurodevelopmental disorder phenotypes. 17 of them affect at least one exon, so exon 7, 8, 9, or the nearby. But most interestingly, 7 out of 14, so 30% of the NDD cohort are purely intronic deletions. That means they do not affect any exon. This is even more important when you consider that 3 of them are purely intronic and the patient doesn't have any other copy number variation elsewhere. So from a clinical perspective, this is very difficult to explain because from a medical doctor, the medical doctor is expecting some exon to be deleted so to have the respective phenotype. Here, at least 3 of them, if not 7, they have a copy number deletion inside some introns, and they do not alter any exon. And it is because of this that we were really interested to move on and to continue our research. So what we did, we performed an integrative analysis of multi-omics data. And if we pick up the title of the research article, that means we explain the first part, novel promoter encoding first exon in DLG2. What we did was to collect data from Roadmap Epigenomic project, specifically about histone modification markers. We collected data from ENCODE about RNA-seq data, specifically RNA-seq data of featured brain tissues. Then we collected data from ENCODE about transcription factor binding sites, and finally, CAGE data from Phantom 5 consortium. Now, the CAGE is a specific technology that sequences the beginning of the RNA. So it targets the transcription start site specifically. Combining these 4 large data sets, we were able to study the DLG2 gene and to discover that there are actually 2 novel exons that were not yet annotated in the reference genome. And we call them coding first exon in intron 7, CFE in 7, and coding first exon in intron 8, CFE in 8. Now we validated this result with SwissProt because we had a positive match by blasting our exons, our new exons, to the SwissProt database. And we also found that those new regions are conserved in mouse. Now I will move to provide a little bit more of technical details and deeper information about the methodology. So we have on the top the DLG2 gene with its exons. We have on the top also the patient cohort. And what we did was first to use epigenomic roadmap to identify some isomodification markers, if present. And as you can see in this picture, we investigated 6 isomodification markers, 4 that are known to be activators, and 2 that are known to be repressor in literature. What is interesting to see at this point is that there are 2 peaks on exon 7 and exon 9 of H3K4 trimethylation, which is expected because at this point, at this location, 2 isoform stars of the gene. What was very interesting to see is that there are also 2 peaks of this H3K4 trimethylation on the introns. And we named those peaks H3K4 peak in intron 8 and H3K4 in intron 7. If we investigate further those regions, we can see that roadmap epigenomic predict that those regions are correlated with promoter activity. And this is predicted in fetal tissues and adult tissues. On those regions, we found CAGE peaks. So activity or transcription start size. We found some transcription factors. And then very interestingly, we found also from RNA-seq data, we found transcription activity. What you see here is the coverage from RNA-seq file downloaded from ENCODE. Now, the vertical dotted line represents the splice site, while the darker region represents the protein match by blasting the exon, by blasting the region to SwissProt. If we zoom in a little bit, we can see the exact coordinate of the splice site, plus the canonical dinucleotide for donor splicing site, that is AGGT, plus below the protein sequence, at least the first amino acids. Finally, at the bottom, a little analysis about the conservation using SIGA database, and we found that those regions are pretty well conserved across vertebrates. So this gives an overview a little bit more technical about the methodology that we use. And all these helped us to identify two novel exons validated using each Swiss plot that are conserved in mouse. Now, this is a picture from the article that summarizes what we have done, and that we extended this research to the mouse genome. So basically, we found that those two novel exons in human have the orthologous on mouse. One part of the research that I would like to emphasize at this point that I think is very interesting for the audience is the following. We studied 6 histone modification markers across 4 points of the development of the individual, from stem cell, which is the first line, to neuroprogenitor cells, so the point where the cell starts to become a neuron, then when it is fetal brain, and when it is adult brain. What we can see from this picture is that the epigenomic signal on the 2 non-exons and on the 2 novel exons is increasing over time. And we provide also negative control from a mesenchymal cell, so to show that evidence that there are novel promoters and novel coding exons in DLG2. What we would like to do now is to prove, at least to test, that those regions are associated with developmental delay and intellectual disability. So what we did, and if we pick up the title of the research article, we basically focus on the second part of the title. What we did was to transform the information about the copy number deletion in a bar chart that you see below, where every bar is aligned to an exon. This slide uses the known DLG2 structure, so the exons that are known in UCSC right now. And as you can see, around the exon 7 and 9, is an enrichment of deletion. But if we introduce the 2 novel exons, the 2 novel regions, those 2 regions happens to be the most deleted in this cohort. So this raised a bell in our mind. So we decided to go further, and we did some statistical analysis. So we used, as a cases, the deciphered patient, and as a control, DGV. What we did was to perform a Fisher test, studying how many deletions from the 2 cohorts were affecting the exons. And it turns out that there is an enrichment of 6 to 10 to the minus 4. If we do the same analysis considering the novel regions, the HPs, the enrichment is higher. That means the exons are associated with the neurodevelopmental disorders, but the HPs are more associated, are more linked to these diseases, to these disorders. We perform another analysis. We perform the same analysis using an independent cohort. We found in literature one paper that provided a large cohort of patients with global developmental delay and intellectual disability. It also provides the controls patient. And in performing the same analysis on this cohort, it turns out that the enrichment on the exon is not statistically significant, while the enrichment on those 2 novel regions is statistically significant. This was very good news for us because we had from 2 independent cohorts the same message. that the HPs are statistically associated with NDDs. Now we finally performed a third statistical analysis combining the two cohorts, looking for similar regions to the two new ones that we discovered across all the genome. And it turns out that according to our analysis, only our two new regions resulted statistically significant. So, final part is about the clinical consideration. What I would like to say here is that in the cohort of patients, the 29 patients, 33% have NDD phenotypes, while for the others, they had either different phenotypes or it was unknown. Among the NDD patients, there were three categories that were well represented. That is cognitive, behavioral, and psychiatric. While for epilepsy, there was only one patient, so we couldn't say that much about that. And regarding the gender, we measured a male-female ratio of about 3.7. So basically, male were more affected with respect to boys were more affected than girls in this cohort. And below, we also perform a summary about the inheritance type of this of the copy number deletion. So 7 were de novo constitutive or inherited from a parent with the same phenotype. A total of 11 were inherited from a mother with a healthy phenotype or unknown phenotype. And finally, one patient with copy number deletion inherited from a healthy parent, and 10 copy number deletion. For them, we don't have any information about that. So this clinical consideration let us conclude that the deletion of these two novel regions, the HP in 7 and HP in 8, are considered risk factor for NDD, for neurodevelopmental disorders. So, conclusion, I would like to summarize the work. We started with used four large databases. We combined them to understand that there were two novel exons not yet annotated in the reference genome about the DLG2. Those novel exons are coding exons because there is a positive match in Swissport, and those exons are also conserved in mouse. We perform a statistical analysis to assess the association between those novel exons and neurodevelopmental disorders. All of this has to be combined with the clinical consideration. So to conclude, that the deletion of the novel regions or the exons are a risk factor for the neurodevelopmental disorders. And I would like to conclude with the acknowledgement. So this is all the partners that were involved in this project, in this research. And I would like to thank Dennis again for the opportunity to introduce my research to your audience. Thank you very much.